Hello, I'm glad to give a presentation about my poster, Identifying Putative Genes, a biomedical foundation model-based approach for cell type annotation. The study was resulted from a collaboration between IBM Research and Cleveland Clinic. We developed some kind of a new method to rank genes, combining two types of methodologies. One of them is called pre-training, and one of them is called fine-tuning. We are focused here on a single-cell RNA sequencing for target discovery. So in our methodology that is illustrated here in figure 1, basically we took a very large database with over 1 million cells, PanGLODB. We applied to it a pre-trained model, it's called SC-BERT, and we calculated attribution scores for all of the genes. Later on, we applied a second method. We incorporated into this DB and SC-BERT another database coming from the Broad Institute called SCP-259, focused on colon and on UC. And we applied fine-tuning, and again, calculated attribution scores. And then we were able to use these scores to calculate a ratio for each one of these genes. To get the attribution scores, we used a Python library coming from Facebook called Captum. So we ran these two sets of experiments, and by doing it, we were able to look at all of these genes. There are around 20,000. And to calculate what is called the ratio between fine-tuned and pre-trained, considering the attribution scores coming from both of the experiments. And we were able to show or to identify known associations that make sense related to widely studied genes, like this one, MOC2, and ZG16. But we were also able to identify, for example, interesting or potentially interesting associations, or I would say high ratios between fine-tuned and pre-trained to different other genes, considering, for example, REP15. We used two sets of parameters. That's how many cells we basically consider, just to show the differences. And what I'm showing here is basically, I didn't mention before, it's focused on one cell. So in this case, it's a goblet here. When you look at stem cells and you run the same experiment, you see that the ratios are much lower. So the signals are much lower, and it means that stem cells are understudied, at least within the context of colon. So the conclusion is that we were able to come up with a method that takes into account pre-trained and fine-tuned methodologies using Captum and attribution scores to identify potentially interesting genes for a further investigation. And here are the references, the SC-BERT, the SCP-259, and Captum. Thank you very much.